Hello, I'm Srijita Acharya, a doctoral student in the Indian Institute of Technology, Jodhpur, in India. I work in the Structural Bioinformatics Lab headed by Dr. Sucharita Dev. I'm presenting my poster on investigating protein mutational robustness in human health and disease. First, let us get introduced to protein mutational robustness and its relationship with protein quaternary structure. Protein quaternary structure is the native state that a protein achieves by progressive folding, and this architecture is crucial for protein function. For a large number of proteins across many proteomes, the native quaternary structure is multi-subunit, and in many cases it is symmetrical and homo-oligomeric. Due to this high abundance, it is necessary to study the quaternary structure properties and evolution, which occurs by accumulation of mutations. So when a missense mutation occurs in the DNA replication event, it is translated into an amino acid substitution in the protein level. In the next step, there are two possibilities. Either the mutation affects structure and function negatively, resulting in a disease phenotype such as the widely known sickle cell anemia. The other possibility is that the mutation is neutral and it is tolerated without significant damage to the structure and hence function, thus making it a homolog, and this increases the family size and robustness to mutation. So, the consequence of a mutation can be dependent on its quaternary structure properties. We aim to study structural properties and analyze their relationship with protein family size, which is an indicator of robustness or evolvability. So we curated our dataset from 3D complex database, which contains protein quaternary structure information and annotations for structure quality errors as well as other related structural properties. We obtained our dataset of around 10,000 homodimers by filtering for structure quality error annotations as well as redundancy up to a level of 90%. Then we identified and analyzed some structural properties by correlating them with family size, which is calculated by conducting a blast run and counting the number of blast hits with coverage greater than or equal to 80% and an e value of up to 0.01. So far, we have identified properties relating to protein size and shape, protein compactness, and physicochemical properties, as well as its functional parameters. Some of the properties, such as molecular weight, chain length, are indicative of protein size and shape. Other properties, such as residue accessibility, contact density, the extent of intra-subunit non-covalent bonding, as well as the organization of amino acids into secondary structured elements. These are indicative of compactness as well as other physicochemical properties. We also aim to look into functional parameters such as the interactome size, ligand binding sites, and their molecular function. So, when we look into protein size and shape, we observe that family size increases with increasing subunit length and molecular weight up to a certain point, and then it decreases. This indicates that very small or very large subunit proteins are sensitive to mutations and are less evolvable than moderately sized ones. This trend is similar for shape, which is indicated by a score that we call shape index, that is calculated in angstroms, and it's defined as the difference between the closest surface residue and the most distant surface residue from the center of mass of the protein. This value is low for globular proteins and it's high for elongated proteins, and moderate for proteins which are somewhere between globular and elongated, and their shape is mixed. When we look into protein compactness or packing, we observe that with increasing subunit compactness, family size increases, and this indicates that highly compact or packed subunits are more robust and adaptable to mutations. However, that is not the case for interface packing, which is indicated by parameters such as local interface density and contact density of the interface. We have calculated these parameters as the number of atom-atom contacts per residue within a defined area or within a defined space of the protein. When we look into physicochemical properties, they also play an important role in evolvability. We see that the organization of amino acids into different structural elements such as alpha helices, beta strands, as well as loops, in those cases also evolvability or family size increases up to a certain point and then it decreases. Similarly, when we look at the proportions of different amino acid groups, we observe that the proteins with highest non-polar amino acid residues are the most evolvable. When we look at other active amino acids, such as aromatic amino acids or positively or negatively charged amino acids, we see that with increasing proportions of those kinds of amino acids, the family size decreases. We are in the process of identifying and investigating more such structural properties as well as functional properties. So, what we can conclude from it is that structural properties play an important role in protein evolution by influencing the molecule's capability to tolerate mutations. We aim to identify more such properties and correlate these properties amongst themselves as well as with family size. And this process would help us to build a predictive framework for assessing the effects of missense mutations based on structural properties. Thank you.